Welcome to The Explainer. Today, our mission is super clear. We are going to demystify the abstract, microscopic world of genetic engineering. I know this topic can feel incredibly complex when you're just staring at a wall of text in a textbook, but by breaking down the mechanics visually, step by step, we're going to help you confidently ace your exams and truly understand the actual machinery of life.
Okay, let's dive into this. Have you ever stopped to wonder how we actually get the insulin that millions of diabetics rely on every single day? Did you know scientists literally create it using bacteria? Honestly, it sounds like straight-up science fiction. But as we visually map it out here, you'll see exactly how cutting, pasting, and transforming genes makes this modern medical miracle entirely possible.
All right, here is our roadmap for today. We'll kick things off with the insulin miracle, explore DNA as our master blueprint, and decode the triplet code. Then we'll map out the actual engineering process, hit some common exam mistakes so you don't lose easy points, and finish up with a final review.
Let's start with Section 1, the insulin miracle. We're looking at the real-world impact here.
You know, grasping genetic engineering isn't just about passing a test. It is fundamentally crucial for the evolution of modern medicine and agriculture. But look, from a student's perspective, mastering these exact mechanics could literally be the difference between a B and an A on your next exam. So let's make sure you get that A.
Moving right along to Section 2, DNA, the blueprint. Time for some foundational biology.
Let's zoom right in and magnify our view. At the structural level, we have DNA, or deoxyribonucleic acid. This is a complex double helix molecule made of repeating units called nucleotides. I want you to really visualize that classic twisted ladder structure. It acts as the exact physical foundation of our biological blueprint.
Now, what's really interesting about this slide is how we can break that structure down even further. Every single nucleotide is composed of three exact parts: a phosphate group, a sugar called deoxyribose, and a nitrogenous base. These three components physically lock together to form the fundamental building blocks of life. And if we focus specifically on that nitrogenous base, well, there are four variations that make up the actual rungs of our DNA ladder: adenine, cytosine, guanine, and thymine, or as you'll usually see them, A, C, G, and T. These four bases represent the entire alphabet of our genetic code. It's kind of amazing when you think about it.
So, with all that in mind, what actually is a gene? Well, a gene is simply a specific, isolated segment of DNA located on a chromosome. It carries the explicit instructions for making proteins. So we're essentially bridging the macro scale of the entire chromosome right down to a specific, highly functional microsegment.
Let's move to Section 3 and see how this builds. Decoding the triplet code. How do we actually read these bases?
Biological instructions are written in what we call a triplet code, also known as codons. As you can see, every combination of exactly three bases mathematically maps to one specific amino acid. It's a precise one-to-one calculation that the cell executes to string together a protein. Think of it like a biological translation dictionary.
All right, Section 4, the engineering process. This is where we get into the active, direct manipulation.
Genetic engineering is the direct manipulation of an organism's genes in a lab. But to move a gene from organism A to organism B, we can't just toss it in there. We need a vehicle called a vector. In bacterial engineering, we typically use a specific circular piece of DNA called a plasmid. Think of the plasmid as the literal delivery truck for our desired gene.
Now, the engineering process follows a strict step-by-step chronological flow chart. Follow along with me here. Step 1, we isolate the desired gene. Step 2, we cut the DNA to create open ends. And Step 3, we physically paste that gene into our vector plasmid. Memorize this exact progression and you've completely mastered the underlying logic of the process.
But wait, how do we actually do the cutting in Step 2? We use restriction enzymes. You can think of these as biological scissors. They cleave the DNA sequence in a staggered way, which leaves these exposed, overhanging sequences that we call sticky ends. These sticky ends are super important because they allow the new gene to easily slot right in and pair up.
Once those sticky ends have found each other, though, they aren't permanently attached yet. Enter our next tool, DNA ligase. And this brilliantly illustrates how it acts as the biological glue. It permanently joins the DNA fragments together, sealing the newly inserted gene seamlessly right into the plasmid ring.
Moving on to Section 5, Common Exam Mistakes. We absolutely need to help you avoid these common pitfalls.
So the crucial point is understanding this exact difference. Genetic engineering involves the direct mechanical manipulation of genes in a laboratory setting. Selective breeding, on the other hand, relies purely on natural variation and mating over multiple generations. Do not mix these up on test day.
And here is another massive pitfall: swapping your enzymes. Remember, restriction enzymes equal the scissors. They cut the DNA. DNA ligase equals the glue. It joins the DNA fragments together. Burn this side-by-side comparison into your memory so you never ever confuse the biological scissors with the biological glue.
We made it to Section 6, Final Review and Tips. Let's quickly consolidate everything we've covered. Okay, here's your quick-fire recap. DNA is our genetic double helix blueprint. Genes are specific segments that carry the triplet code to make proteins. And genetic engineering? That uses vectors, restriction enzymes, and DNA ligase to physically cut and paste those genes between organisms.
And before we go, here is an actionable exam tip for you. Don't just passively read the textbook. Practice physically drawing out the sequential flowcharts we covered today. Draw the plasmid, draw the scissors, cutting those sticky ends, draw the ligase, sealing the gap. Recreating the physical mechanics on paper yourself will absolutely lock it into your long-term memory. I'll leave you with this final kind of mind-blowing thought. If we can map out and manipulate this micro blueprint to engineer bacteria that literally manufacture insulin for diabetics today, what incredible, life-saving organisms will we be able to design tomorrow? The blueprint is quite literally in our hands. Thank you so much for joining me in this explainer, and I'll catch you next time.